This is Microbiology, Lecture 15. We're going to talk about the mechanisms of microbial pathogenicity. Now, pathogenicity is the ability to cause disease. If we can understand a microorganism's ability to how the mechanisms involved in how it causes disease, its mechanisms of pathogenicity, then that can give us an appropriate target in order to attack it and kill the microbe. And it could also, or it might, if not, then it might give us a way of avoiding, at least avoiding infection. So these are major bonuses of trying to understand the system, the way in which the microorganism, what mechanisms it uses to cause disease. The extent or degree of ability of an organism to cause disease is its virulence. Now, when we look at mechanisms of pathogenicity, there are certain key phases that need to be studied. One are the portals of entry, understanding where the organism, how the organism gets into the body, how it penetrates or evades host defenses, how it causes its cytopathic effect to damage host cells, and then, and this is important for every organism, how it gets out, because it has to get out of the host. It has to have some sort of portal of exit in order to infect another individual, so that it can keep on alive, so it can stay alive as an organism and multiply. So we're going to go through each of these. These here are additional factors that need to be considered, and we'll mention those, but the four key things are portal of entry, penetration or evasion of host defense, cytopathic effect or damage, and portal of exit. In terms of portal of entry, because epithelial tissues cover all surfaces, dividing the outside world from the body tissues, portal of entry has to somehow be through an epithelial membrane or surface. Pathogens do not all get into the body the same way. There are different avenues or portals of entry. One way is through a mucous membrane, and mucous membranes or mucosal epithelia line the respiratory, GI, GU tracts, and also the conjunctiva of the eye. The surface layer of the eye is an epithelial mucosal membrane. Now, the most common portal of entry or entry point is by inhalation into the respiratory tract. Remember that the respiratory tract, the surface going down, is a pseudostratified columnar epithelium with cilia on it. It's a ciliated pseudostratified columnar epithelium. And then once you get into the respiratory zone, you have the alveoli surface, which is made up of squamous epithelial cells. It's a simple squamous epithelium. They're called type 1 epithelial cells, and they line that. So the most common point of entry among organisms is the respiratory tract. Certain organisms will enter through the respiratory tract, quite a large number of them. Another common entry point is through the GI tract. After eating of contaminated water or food, or kids sticking their fingers, and adults sticking their fingers in their mouth and ingesting things that they've picked up, touching surfaces all around. Some of the organisms that can enter through the respiratory tract include rhinoviruses that which are a major cause of common cold. There are other causes, but rhinoviruses are a major one. Influenza virus, which causes flu. Mycobacterium tuberculosis, which causes TB or tuberculosis. The virus that causes measles. It's incredibly highly virulent, easily transmitted. If there's one person in a room, then you'll get, in an enclosed space, then you'll get infection. And if people aren't vaccinated against it, then you'll get infection of 90 to 95% of the people in the room after they've shared the airspace for a fairly short period of time. Also, coronaviruses, SARS, COVID-19 have used the inhalation entry point through the respiratory tract. Some of the organisms that gain entry by GI include poliomyelitis virus causing polio, hepatitis A virus, typhoid fever, the bacteria that causes typhoid fever or cholera. SARS also can enter through the GI tract, as can, by the way, COVID-19. I didn't put it in. And, of course, Clostridium difficile. There's also some possibility of entry through the skin, the stratified squamous epithelium that covers the outside of your body. Again, it all depends on the organism, which is its favored portal of entry. Many organisms getting onto the skin have no effect whatsoever. And for many organisms, they have multiple possible routes of entry or portals of entry, but they don't necessarily cause the same degree or characteristics of the disease, same kind of set of signs and symptoms if they enter through a different portal. In terms of the skin, there are entry points through sweat glands or hair follicles that allow for entry of some organisms. There is also the parenteral route of entry. It's a term that you may not be familiar with, but is important. The parenteral route refers to piercing through a surface, directly through that surface membrane, through an epithelial membrane, via a break in the squamous or mucosal epithelium because of injury or penetration. You know, like a needle cut, something like that. Tissue injury. Okay, so as I said, many organisms have a preferred portal of entry. They can gain entry into the body by different portals, but may only cause disease if they enter by the preferred portal. An example of that would be Salmonella typhi. The organism is a bacteria which causes typhoid fever. If swallowed, typhoid fever will, and if you know, for every organism there is a certain number that you need in order to get infected. But if swallowed, and it doesn't take much, if you swallow it, it'll cause typhoid fever. If rubbed into the skin, nothing. Okay, so we've touched on, I just mentioned, and the number of invading organisms is very important. You know, the degree and how quickly the infection, you get an onset of symptoms can change with the number of organisms that enter into you. Sometimes there's a critical threshold. Your body can avoid infection at a lower number, but once you pass that certain threshold, then you do get the infection. Also, adherence is very important. Microorganisms, all viruses, bacteria, they always have some molecule that they'll adhere to. It's quite specific. For example, with the SARS-CoV-2 virus, which causes COVID-19, it will adhere to a molecule found quite commonly found at a high density in the respiratory tract. It's called ACE2. It's a molecule on the surface of the cells, and it'll adhere and enter following adherence. But ACE2 actually is found in other parts of the body, many other tissues, particularly in the cardiovascular tissue, in the GI tract also. So the virus can have effects at a distance, or it could enter through the GI tract. For example, the point I'm trying to make here with COVID-19 or SARS-CoV-2 virus, if it enters into the lungs and it binds to its target that it can adhere to, the ACE2 molecule, then it can enter into cells, and if enough gets produced in those cells that it gets into the bloodstream, then it can also spread through the blood and infect cells lining the vascular system, because they also express the ACE2 molecule. So adherence and number of invading microbes is also an important consideration when you get entry through a portal. Exposure, therefore, as I have been talking about, the exposure is not the only determining factor of whether a person gets a particular disease. The number of pathogens entering is important, and that can be demonstrated that quantitatively by accurate number by the fact that every pathogen does have a characteristic measures of pathogenicity called the infective dose 50, and also ID50 and also LD50. If you look at a population, and when you're talking about test populations, you're talking about a animal model that's used, an animal that is used in labs that can catch the disease, the infection, and has similar responses. If you look at animals, there'll always be a certain range in number of organisms that can infect 50% of the animals. If you start at a low enough number, none of them get it. If you raise the number that you give a group of animals, maybe 10% get them, and at some point you'll get to an ID50 where 50% and above get the infection, and that's the ID50, infective dose 50%. And an important thing to understand, it helps understand, is the virulence of the organism, how easily it can infect. LD50 tells you its level of pathogenicity in terms of what does it take to kill 50% of the test population. And here is an example of an organism. Now we're back to the idea of portals of entry and multiple portals of entry. Some organisms, as I told you, can enter by several different routes. An example of that would be Bacillus anthracis, an organism that causes the disease anthrax. It is a endospore forming organism. It's found in the soil contaminated with feces quite often, and if the portal of entry is the skin, then the infective dose, 50% infective dose, is 10 to 50 endospores. As few as 10 to 50 endospores in the skin will cause an infection. Inhalation requires more for 50% of the test population to get infected, and ingestion takes even more, quite a bit more. There are, however, differences in terms of the nature of the disease that is induced by depending on where the portal of entry is. Okay, let's go to the next point, subtle or contributing factor in terms of portal of entry, and that is adherence. Pathogens, as I said, need to adhere to the host cells in order to infect, and they will adhere by binding to a particular molecule. The molecule acts as the receptor on the host cell surface, and the organism has the ligand molecule on its on it. Remember, binding in biological systems is often specific and involves a ligand molecule and a specific receptor molecule. So on the pathogen you have the ligand on the pathogen, and on the target cell that can get infected you have the target molecule acting as a receptor. Okay, and as I said, for the SARS-CoV-2 virus, the receptor on human cells is the ACE2 molecule. ACE stands for angiotensin converting enzyme. This is not the same one that you learned about when you learned about the renin-angiotensin system, where angiotensin 1 is converted to angiotensin 2 in the lungs by angiotensin converting enzyme. That is ACE1. Here we're talking about a similar enzyme, but it is ACE2. That ACE1 didn't really show up at all, did it? Let's get rid of this. Okay. With bacteria, you can have types of molecules, ligand molecules that are called, in general, the general name is adhesins. They will bind to receptors in the host cell. The adhesin may be part of the bacteria's glycocalyx, or it may be on the pili or the fimbria or flagella. And here's some examples in black ink, so they're just for your own interest if you want. Adherence really is an important part of the life history of microorganisms that remain on surfaces exterior to a body, but are important in terms of healthcare. They form what are called biofilms. So many bacteria actually are able to form biofilms on moist surfaces. If the surface has some organic material, organic molecules on them, then you can get bacteria in that material, and it can build up several layers thick, and it can also get deposits of calcium that make it quite tough material. This is what forms on your teeth, and what forms as plaque on teeth and has to be removed when you get your teeth cleaned by a hygienist. But these kind of layers can also quickly form on catheters or vascular stents in a person's vascular system, in heart valves, contact lenses, etc. Biofilms provide excellent protection for the bacteria against any disinfectant or antibiotic. They really protect them. They're sequestered underneath. They're hidden and protected from attack by being underneath that biofilm, and really take quite an effort to get rid of. Okay, let's move on now to talking about penetration or evasion of host defense. Penetrating is not having a portal of entry is not enough. The organism still has to get around the host defense and spread within the body. One way of inhibiting and avoiding host defense mechanisms, one of the very earliest thing whenever something foreign, like a microorganism, or it could be anything, even something that's not alive, enters into you, that one of the first things that happens is that phagocytic cells move towards the object. They leave the bloodstream, move towards the object, accumulate there. They're active. They are producing quite toxic molecules. They'll phagocytose and try to kill the organism. So quite a few bacterial organisms are able to produce a capsule on the outside. And that capsule will impair phagocytosis. It'll inhibit phagocytosis by host cells and therefore increase the ability to cause disease, increase the virulence of the organism. Sometimes the organisms have certain cell wall components that protect them. For example, the wax in Mycobacterium cell wall will resist hydrolytic enzymes in lysosomes. Phagocytic cells like macrophages or neutrophils can come along and phagocytose a bacterium, and they'll take them into a membrane-bound phagosome, And then a lysosome with lysosomes with containing hydrolytic enzymes will fuse with the phagosome, and you get a phagolysosome, and it'll normally break down the bacteria. But if it's a bacterium like Mycobacterium leprae or tuberculosis, then it'll be protected by the wax in the cell wall. Some organisms are able to produce enzymes, and those enzymes help them protect them or help them move quickly and spread easily, penetrate new areas. Some examples of that would be an enzyme in some bacterial organisms called coagulase, which can cause formation of fibrin that surrounding the a site of bacterial infection, and that helps wall off the infection and prevents immune defense systems from, like phagocytic cells, from attacking the bacteria. Other organisms may produce collagenase, which can help them penetrate through connective tissue and move quickly forward. And others produce streptokinase, another enzyme that helps dissolve clots. After all, when you have an injury, then you're going to have also the potential that bacteria can get into the wound. It may have happened at the time that the wound occurred or just after. But the bacteria are in there and you have injury, so you have bleeding and you have blood clots. And the blood clots normally help wall off the area and prevent further spread of the bacteria. But some organisms have the enzyme streptokinase, and it can dissolve a clot and help it spread further. Another type of mechanism that organisms use to evade or penetrate past host defenses, and this is quite a interesting complex mechanism. It's called antigenic variation. They have surface proteins on them which can trigger a specific immune defense mechanism in an infected individual so that their immune system attacks them. But some organisms, and the best known example, the most studied example of this is the organism that causes malaria. It's a protozoa called, the genus name is Plasmodium. There's several species that can cause malaria. There's Plasmodium vivax and Plasmodium falciparum are two examples. What they have are the ability, the genetic-based ability to change the proteins that are expressed on the surface. So if they've infected an individual, the individual may develop antibodies or a T-cell defense against that, targeting that protein. But if they change the proteins expressed on the surface, then it's like a whole new organism, totally different, that your immune system has never seen, and they can do this multiple times. Another mechanism that organisms use for penetration host defense is they can alter the host cell cytoskeleton and help bacteria enter into the cell. Remember that intracellular organisms are somewhat protected by being able to survive intact inside a host cell and not killing off the host cell. Okay, let's move on to damage to the host cell, the cytopathic effect, the injury caused by organisms. There are a number of different ways in which organisms can injure cells. Let's go through these. Once bacteria enter and get past the first line of nonspecific defense, and that's the very first line, most nonspecific defense mechanism actually is the epithelial barrier between the outside world and the tissues, and bacteria have to get past that, and once they're in there, then they have to evade the second line of nonspecific defense, which is also called the innate immune system. All right. So this is a non-specific defense. The innate immune system, and that includes phagocytic cells, the inflammatory response, antimicrobial peptides, small proteins. And once they evade that also, then they can start causing, then they'll, if they manage to avoid those, then they'll, multiply and cause injury or damage to host cells. And that injury can occur by four main methods. It depends on the organism. The organism may use up host nutrients or cause direct injury to the immediate area. They may produce toxins which spread through the body and have quite serious effects. Depending on which toxin, they'll have different effects. Or they may actually induce a hyperreaction by the body's immune system, and you get a hypersensitivity reaction. We'll go through these one at a time here. Using of the host nutrients, some bacteria have the ability to take up and sequester away from, hold and keep away from the host some essential nutrients, for example, iron. Quite a few bacteria produce what are called siderophores. They can form complexes with iron, and they'll even can remove the iron from host proteins that need iron to be able to be functional. The siderophore iron complex gets taken up by the bacteria, and the bacteria use that iron for their own purposes, denying the host cells from having iron available. Direct injury in the immediate area. Some pathogens will enter host cells and use them as a nutrient source and produce some waste products that often lyse the host cell. That's how they trigger their cytopathic effect, and they leave and infect other cells. Some pathogens do something quite different. They will induce the host cells to take them up and are safe within the cell, so they can remain safely inside the cell itself, hiding from the defense mechanisms of the host, and multiply within those cells without lysing. Now the third topic is a large one, and it refers to the production of toxins. And we've already talked about this a little bit when we talked about exotoxins and endotoxins, and here I'll go into somewhat more detail. Toxins can be released by microorganisms pathogens that have infected a person, and they're often the cause of most of the injury that occurs in the host tissues. They can be released. They're soluble. They'll spread throughout the body by being transported in the blood or through the lymph, which drains the tissues of its extracellular fluid and returns it back to the blood. They are the cause of, depending upon the organism, can cause fever, diarrhea, vascular changes. They can cause tremendously severe, quick acting responses. Once released, they are really extremely toxic molecules. They can cause types of paralysis at different nerve endings, vascular changes, pressure changes, and can lead to shock and multi-organ failure. About 40% of toxins, roughly, will cause damage to the eukaryotic cell membrane. If the toxin is in the blood, that is, of course, called toxemia. In general, in terms of bacterial infections that can are pathogenic organisms that cause disease, there are two general types of toxins. There are exotoxins, which are produced by gram-positive organisms, a few gram-negatives, mostly gram-positives, vast majority are from gram-positives, and endotoxins, which are produced by gram-negatives in that they're not secreted like exotoxins, but they form part of the cell wall of gram-negative organisms. When you look at bacterial toxins, they have a very low LD50, a really low 50% lethal dose, a dose that will kill 50% of experimental group of animals. Many toxins, and here we're not talking about endotoxin, but exotoxins from gram-positives, can be inactivated and used in a vaccine to trigger a specific immune response. It's not to the bacteria, it's to the toxin that the bacteria produces. So if you've been vaccinated with a toxoid is an inactivated toxin. They purify toxin and then inactivate it and then use it in the vaccine. Then you have immunity to the toxin itself. And that's how some vaccines work. They don't stop you from actually getting infected with the organism, but they will block the organism's ability to cause damage to your cells. A good example of that would be the vaccine used for Corynebacterium diphtheria, the organism that causes the disease diphtheria. You get, when you receive the vaccine against this, you're getting inactivated toxin or toxoid, diphtheria toxoid. Your body will respond by producing antibodies. So these antibodies are called antitoxin or antitoxin antibodies. Okay, and they're always antibodies molecules are always very specific. In terms of exotoxins produced and secreted by gram-positive organisms, they simply are part of the normal growth and metabolic activity of the cell. Some, and we'll get into that later, but I'll just mention it, right? I'll get into more detail later, but I'll just mention that some bacteria pick up the ability to produce exotoxins by being themselves infected and transduced by bacteriophages, viruses that can infect bacteria. So they secrete as part of their normal metabolism, they'll produce and secrete the exotoxin. It gets released also, if the bacteria gets lysed, it'll also get released, but normally they're just secreting it. The source of endotoxins, as I said, are mostly gram-positives. A few gram-negatives can produce exotoxin. There are many different specific exotoxins characteristic of different bacterial strains or bacterial species. Again, it's a byproduct of the growing cell. Plasmid genes often carry the gene for the exotoxin. Sometimes they pick up the ability because of bacteriophage transduction. These exotoxins are proteins which often have enzymatic activity, and characteristically they do not cause fever. They can be neutralized by antibodies against the toxin. Often the toxin is used in the vaccine in those cases, so they can be neutralized by antitoxin. The LD50 is particularly tiny, very, very tiny, especially if they have enzymatic activity. The disease associated with bacterial organisms, gram-positive bacterial organisms that produce an exotoxin, is often, the disease process is often, the damage to cells is often caused by the toxin only. Nothing else, just that toxin. It's not some other product or the way they grow into tissues, just the toxin. The signs and the symptoms are caused by the exotoxins. For example, botulinum exotoxin produced by the organism Clostridium botulinum is extremely toxic, and even and toxicity, by the way, is different for different animal species. Humans actually are not as sensitive as guinea pigs. Guinea pigs are a very good animal model for disease caused by botulinum exotoxins. The LD50, lethal dose that will kill 50% of a population group of guinea pigs tested with it, is 10 to the minus 9 grams. That's 1/1000th of a gram is enough to kill. If you give each animal in a group, say you inject 20 animals with 1/1000th of a gram each, 50% will die. Examples of diseases that are treated by vaccinating with toxoids or inactivated exotoxin include diphtheria, tetanus, and botulism. When you look more closely at exotoxins, you can divide them up into 3 separate groups: type 1, type 2, and type 3. Type 1 are superantigens, and I'll explain what that means in the next few slides. Type 2 are membrane disrupting toxins, and type 3 are AB toxins. The type 1 toxins or superantigens, they have an ability to stimulate in a non-specific way T cells. So they'll, you have a large number of T lymphocytes or T cells in your body, and each T cell has a specificity for different foreign structure. It has a receptor for a foreign molecule. It's part of your immune system that helps you defend you against different specific foreign molecules that could get into you, including the ones that are on microorganisms. And typically, if you get an infection, then a small number of T cells that can respond to that will get activated and multiply into a large number and therefore help defend you. The thing about superantigens, unlike other foreign molecule or antigen molecules, is that they'll activate a very, very large number of T cells. It doesn't matter what the receptor is on the T cell, it'll get activated by these superantigens or type 1 toxins. And when they get activated, they release cytokines. And cytokines mediate or trigger different kinds of fairly strong immune responses. But here you're getting such a large number of T cells activated that you get a very large amount of cytokines released. So you get an intense immune response that's so potent that it triggers high fever, nausea, vomiting, diarrhea, shock, and even death. The injury is caused by the inappropriate, overly large scale of activation of the T cell defense mechanisms. It triggers huge release of cytokines, triggering other kinds of defense mechanisms, but it's such an inappropriately large response that it's actually the body's own response that got triggered, inappropriately large response that actually causes the injury to the person. That's superantigens, also called type 1 toxins. An example of that would be the toxin associated with toxic shock syndrome, for example, which is caused by group A staphylococcal organism gas. I said staph pyogenes, but it's Streptococcus pyogenes. Group A Streptococcus organisms can produce superantigen response because they have an exotoxin that can have a superantigen effect. Okay, let's talk about type 2 toxins, another type of exotoxin group. These are membrane disrupting toxins. They have the ability to lyse the host cell's, damage the host cell's membrane by inserting protein channels into the plasma membrane and therefore causing lysis of the cell. One example of that would be hemolysins that are produced by both streptococcal and staphylococcal organisms. An example would be Staphylococcus aureus exotoxin. Some Staphylococcus aureus organisms are pathogenic, will produce an exotoxin. Another example would be Clostridium perfringens exotoxin. Clostridium perfringens is the organism that can cause gas gangrene, and that also has the ability to disrupt or damage the plasma membrane. It disrupts the phospholipid bilayer. AB toxins, or type 3 toxins, have a different approach in the mechanism by which they damage tissue. They are made up of two polypeptide chains. There is an A chain and a B chain polypeptide. They're proteins, so they have two chains held together by covalent bonds between the two chains, usually disulfide bonds between the two chains. The B chain will bind to the host cell, so it allows the toxin to attach to the host cell, and the A chain is the toxin. Okay, so B part is for binding, and the A is the toxin. Typically, it'll inhibit a certain protein synthesis and kill the cell. There are many exotoxins, as I told you, that are produced as a result of phage transduction, lysogenic insertion of the prophage gene into the into the cell genome, or I'm sorry, into the bacterial genome, giving it the ability to produce the toxin. An example of that would be diphtheria toxin, which is coded for by the tox gene. There are some toxins associated, it's called erythrogenic toxins. They'll damage blood capillaries and cause a rash in the skin. Things like streptococcus or staphylococcus organisms may have this. Scarlet fever caused by streptococcus pyogenes, which is the same, again, I remind you, as Group A Streptococcus organism. Group A Streptococcus organism, Streptococcus pyogenes. Many of them can produce these erythrogenic toxins. Botulinum toxin, which is, of course, the toxin associated with Clostridium botulinum. This toxin acts as a neurotoxin. Botulism is food poisoning. All the Clostridium organisms, Clostridium botulinum, Clostridium perfringens, Clostridium tetanus, Clostridium difficile are anaerobic organisms, are anaerobic spore-forming organisms that can produce toxins. This one for Clostridium botulinum acts at the neuromuscular junction, causing paralysis. The tetanus toxin caused by Clostridium tetanus is also a neurotoxin that causes and triggers muscle contraction. It also acts at the neuromuscular junction, but instead of causing a paralysis, causes muscular contractions, which is the reason why, rather, the disease also happens to be called, the layman term for it is, tetanus is lockjaw, because one of the first symptoms, or I should say one of the first symptoms, is that the person's jaw starts to lock, so they have trouble speaking because their jaw muscles are contracting. So they have their jaw is locked shut. Eventually, and fairly rapidly, if untreated, it would progress till all the muscles were tensed because of its neurotoxic effect at the neuromuscular junction. Vibrio enterotoxin, the toxin associated with cholera. Vibrio cholera is the organism that causes the disease cholera. So Vibrio enterotoxin, it's an enteric intestinal infection, will cause the loss of fluid and electrolytes, causes very, very severe diarrhea. Staphylococcal enterotoxin can also cause loss of fluids and electrolytes. Many of the organisms that infect the GI tract will actually cause fluids and electrolytes to instead of getting absorbed in the gut, will move into the gut space and therefore cause severe diarrhea. Okay, as I told you a bit earlier, many of the exotoxins are in fact proteins produced by genes, and they're a gene product, and for many organisms they actually have the gene because of lysogenic conversion. They were infected with a bacteriophage that had a lysogenic cycle, and the phage or prophage DNA is inserted into the bacteria's chromosome, and the bacteria picks up the ability to produce exotoxin. That's true for Corynebacterium diphtheria, which will form diphtheria toxin. That's due to lysogenic conversion. Streptococcus pyogenes, a group A streptococcus or gas organism that causes a number of different types of infections. Clostridium botulinum, Vibrio cholera. All of these are due to lysogenic conversions, giving them the organism the ability to express these exotoxins. Okay, let's move on to talking about endotoxins. Endotoxins are a product of gram-negative bacterial organisms. However, they are not secreted. They actually form part of the cell wall of the gram-negative organism. They are released into a person when the bacteria dies. And when you have an infection, the bacteria are growing rapidly and dividing, but a few of them are still also dying. A significant number are dying at the same time, but overall it's increasing as the infection gets worse. But still, you're going to have endotoxin getting released with time, and the endotoxin is that part which forms. It's not very well drawn here, but it forms quite a significant part of the cell wall all over the cell wall there. It's gonna trigger its effects because one of the characteristic ways that some of the bacteria are dying in you after you get infected is that phagocytic cells move to the site of infection. So cells like macrophages and neutrophils, although a macrophage is probably the more longer term actor. Macrophages phagocytose the bacteria. They take them into phagosomes, which fuse with lysosomes, and you get a phagolysosome. The bacteria gets killed, degraded, and the endotoxin is going to trigger a response in the macrophage. It will trigger production of a cytokine called interleukin-1. And the interleukin-1, this is a product from the macrophage itself. It's a normal response to the presence of endotoxin. If macrophages are exposed to endotoxin, they will produce IL-1. The IL-1 gets released, goes to the bloodstream. It'll trigger the temperature setting area of the brain in the hypothalamus to raise the body temperature, but it will also trigger other effects besides a fever. It triggers other responses, and this is what starts off the whole cycle that can lead towards a really high fever. It not only triggers this interleukin-1, but you get other molecules being produced, particularly a molecule called tumor necrosis factor, another one called nitric oxide. And in large amounts with serious gram-negative infections, you can get tissue injury due to an excessive amount of release of these molecules. The reason the macrophages release the molecules is because both TNF and NO, those are the abbreviations for tumor necrosis factor and nitric oxide, both TNF and NO have antibacterial activity. But when released in too large amounts, they will trigger weight loss, tissue injury, hypovolemia associated, drop in volume of the fluid in the vasculature, because it gets leaky and fluid moves out. So you get a drop in blood pressure, and it can eventually lead to septic shock, multiple organ failure, and death. In Canada, there are approximately 30,000 deaths per year due to septic shock as a result of these gram-negative infections. About 50% of the cases die. These people end up in ICU, and it's hard to predict which ones will make it through or not. They're supported, and half of them make it and half of them die. They also often have other underlying diseases, but processes going on. But you know, this can push them over the edge. Septic shock is a serious consequence of gram-negative infection, all due to endotoxin. Again, an example here of a bacterial organism producing a toxin, which, just like in the case of superantigens, type 1 toxins, exotoxin, here with endotoxin, you're getting the body's own normal response being so large here with a significant infection that it actually causes injury and potentially death of the infected person, the patient. So just to summarize, as we did before for exotoxins, the source are gram-negatives. It's present in the it's actually present in the as a molecule called lipopolysaccharide, or LPS. I'm going to write that out for you. LPS is this is endotoxin. Lipopolysaccharide is the same as endotoxin. LIPO, lipo, it's part lipid. Polysaccharide, part carbohydrate, part lipid. I don't know if I have the patience to write this. Lipopolysaccharide. So that is the endotoxin molecule. The toxic part of the molecule, in fact, the toxicity lies in the lipid portion of the lipopolysaccharide. It's called the lipid A part of the lipopolysaccharide molecule. Very, very characteristically, and unlike exotoxins, it does induce fever. That's one of the first things that can be, how its effect can be detected by induction of a fever. There is no antibody or antitoxin that can be raised. You can't make a toxoid from it, so there's no effective antibody. The interesting thing, of course, is that all gram-negative organisms have pretty much the same LPS in all of them. So if you could raise an antibody, it would be effective against all kinds. It would probably be effective against all kinds of endotoxin mediated gram-negative infections, the negative effects of that, but there is no, they've tried very hard and they haven't been able to develop an antibody. The LD50 is relatively large, lethal dose that'll kill 50% of a group of animals, relatively large compared to exotoxins, but it doesn't take much. You know, they've done experiments in humans where they inject small amounts, very small amounts will induce fever. Of course, they don't go to the point of injecting enough to cause damage to the person, but they have done experiments. It's pretty tiny, not as tiny as with exotoxins, but still pretty small. The last thing we'll talk about in terms of mechanisms of how organisms cause disease, or their mechanism of pathogenicity, or the portals of exit. Every organism needs to get out. It needs a portal of exit so it can go on to infect another person. That could be by the respiratory tract, coughing, sneezing up droplets and spreading the disease that way. It could be from feces or saliva in the gastrointestinal tract. The portal of exit could be the GU tract, from urine or vaginal secretions spreading to others, through the skin, from the blood, being picked up by biting insects and carried to another person, or like with some protozoal infections like or viral infections like West Nile virus, or protozoal infections like malaria, could be picked up by needles, sharing of needles, syringes. That's true for hepatitis B certainly, and HIV infections. So there are a number of different portals of exit by which an organism can exit and go on to infect another individual, which of course is an absolute necessity if it's going to continue to exist. So it's an absolute part of its life strategy. So all of these are important aspects. All the things we've discussed are important aspects of how an organism causes the disease process, its mechanism, pathogenicity. Portal of entry, including number of invading organisms and ability to adhere, penetration or evasion of host defense, damage, cytopathic effect, and portal of exit. And again, I point out to you that the importance of understanding a microorganism's mechanism of pathogenicity is that it can give us a target to try and attack it, to target a weakness in it. If we can pinpoint one key mechanism, then we can target and attack that microorganism, prevent infection, or at least at the minimum understanding a way of avoiding infection, if you understand portal of entry, portal of exit, etc. So that's it for lecture 15, microbiology, mechanisms of pathogenicity, a very important concept overall.